Big news, the FDA is making it easier to edit people's DNA. Let's talk about it. So historically, FDA approval for new medicine has involved multiple phases of clinical trials. There's multiple ways to do these trials involving healthy volunteers, placebos, patients, etc., but the gist is that you are doing multiple rounds of very thorough testing on as many people as possible. But DNA editing isn't like traditional medicine. In most cases, you're not making a generic drug that can be broadly applied to most, if not all, patients with a specific illness. Instead, you're designing a custom molecule called a guide RNA that precisely matches the mutation in an individual patient's DNA sequence down to the letter. So genetic diseases are typically characterized at the level of the gene, but genes are made up of thousands of individual DNA building blocks, and a mutation of any one of those could potentially give you the disease. So oftentimes, hundreds of people with the same genetic disease can each carry a unique mutation, which means that each of them would need a unique guide RNA. And doing hundreds of simultaneous multi-phase clinical trials on one disease is just not really feasible, especially on rare diseases where only a handful of patients have each mutation. So the FDA has introduced what is called the plausible mechanism pathway. It works on 5 basic principles. First is diagnosis. So instead of taking a cluster of symptoms and trying to figure out which diagnosis best explains all of them, doctors instead look directly at a patient's DNA and identify the specific mutation that is causing the symptoms. Number 2 is the medicine itself. So again, in the case of DNA editing, you're not designing a pharmaceutical drug with broad applications like a steroid or an antibiotic. You're designing a guide RNA that uniquely targets the individual mutation that's making somebody sick. Number 3, the disease has to be well understood. You need to be absolutely certain that A, there are no other options for treatment available, and B, if nothing is done, the patient is going to have an unacceptably low quality of life, if they live at all. Number 4, you have to show that it works in the lab, so you don't get to skip testing it entirely, you just get to skip the part where you test it in humans. And number 5 is proof of clinical benefit. Now this is a little odd because it does require one person to go first. Essentially, they're treating individual patients as miniature clinical trials so that they can figure out if patients with the same mutation should be allowed to get that medicine in the future. And instead of requiring 2 patients, so one to receive the treatment and one to receive the placebo, you can compare an individual patient to themselves before and after receiving the treatment. Now this is only going to be done in rare and extreme cases, so it's mostly intended for babies and very young children who have rare diseases and no other options. And if you want an example of how it's actually going to work, my video on baby KJ would be a great place to start. Researchers like me who are studying the use of DNA editing in medicine have been hoping for years that something like this would happen. And I am so grateful to the researchers, the medical professionals, the policymakers, and especially the patients and carers who have put in the hard work to make this a reality.